All right, so let's go ahead and start. We have office hours. My office hours are Mondays from 12 to 1 p.m. That's you're more than welcome to stop by before the test, and if something here doesn't make any sense, you can always ask me. I'll be more than willing to help. Wednesdays, my office hours are 4 to 5 p.m. Again, same thing. Any office hour time, you can stop by, ask questions. The second we upload the tests, you can come the next office hour that I have. I will have the whole box of all the tests, so you can view your test, and I'll be located in SC 294. Hopefully this is the last review video I'm going to make, because after this I believe we're going to have an SI, so you guys can go to her. I believe her name is Alexis, so you guys can ask her questions. But you can always come to me and ask questions. I just won't be doing this tedious video making process where it looks like I'm just talking to myself. Anyhow, if you're going to come review your test, in addition to taking the test, you're going to need your student ID. During review, I'll exchange your test for your student ID, and then after I'll give it back to you. So when you're taking the test, remember you're going to need your student ID, you're going to need a number 2 pencil, and you're going to need a blue Scantron. Last time a lot of people made mistakes on filling up their names in a blue Scantron for some reason. So remember that first you have to put your last name, and then you put your first name after you put like a space in the middle, okay? So when you put the space in the middle, please make sure to bubble in this region right here. This acts like a space bar. So if you just put your last name and first name and bubble in just those, everything comes together like a mesh of letters, and then I would have to go and see where your last name stops and where your first name. So it's just super tedious, and the longer it takes me to do it, and the more manual work I have to do, the more likely it is that I make a mistake. So in order to prevent that, for your own sake, please make sure to follow that instruction. So yeah, and also make sure you bubble in every letter, right? Because if you don't, the machine's not gonna be able to read this portion here. This portion here is just for you, just to know where to bubble in what. Also make sure to bubble in, if you have version 2, under the special codes, just if you have version B, you put 2, and then put 2 here. If you have version 1, you just put 1, and then bubble in 1, right? If you have version A. And yeah, good luck with that. Okay, so the first thing we're going to talk about, just a review from last time, are the cytoskeletons. Recall that there are 3 you have to know. There's a microfilament, which are made up of actin, and their main job is for cell movement. There are some pretty cool YouTube videos, like animation-wise, on how these actually work. I don't know if you've ever seen like a neuron, it like moves in certain like directions. If you ever want to watch that, yeah, that's basically the microfilaments that are helping the neuron to move. The next one is these intermediate filaments. These guys are made up of many different proteins, so we're not going to really focus on memorizing their names. The main job of intermediate filaments is for like resistant to like compression and like tension, and so then that's really all you have to know about that one. And the microtubules would be made up of tubulin, and their function is for cell division and intracellular transport. That's intracellular transport, so transporting something from point A maybe to point B, right? So if you want to get like some I don't know, vesicle through the axon, you have these microtubules. They act like train tracks within the cell. But other structures are also made up of these microtubules, and some of which are the centrioles. So this is a centriosome right here, and in the centriosome we have a centriole, and these centrioles, they make spindle fibers. So spindle fibers, and they play a huge role in mitosis, or our cell division. Other structures are going to be flagella and cilia, and recall the flagella is the tail. Our sperm cells have it. Other organisms have a flagella too, but for our body, if you're male, your sperm cells have it. Cilia are these like hair-like structures that are in the trachea, and all they do is they sort of propagate, move the debris or small particles that are not supposed to be there up from the trachea into your mouth, so that you can either swallow it or spit it out. Okay, so that's the job of cilia. They move in this like very synchronized wave pattern and push something up. Smokers tend to have lost like the function, like these cilia don't work as synchronized, I guess. That's why you see like long-term smokers coughing a lot, because they rely on like the force of just air to push that up instead of cilia. So that's one way you can remember that cilia, for now at least, the main location you find cilia in is the trachea, okay? Oh yeah, the last one we have are microvilli. I'm just going to mention these guys because the entire purpose of microvilli is to increase surface area. By increasing surface area, I mean like, okay, so here's your small intestine, okay? And you know, in small intestine you have absorption taking place. And absorption can't take place until like these guys touch the wall, like the food touches the wall. So whatever food that's coming through has to touch like the walls for it to absorb. Well, evolution found a way, and that's to like increase surface area as much as it can. So here you have like the epithelial tissue, right? And on top of these epithelial, I'm just going to draw half of them. On top of them, you have these little microvilli. And these microvilli just add an extension. So these are called villi, and these are the microvilli up there. The microvilli just extend, even make the surface area even greater, right? And so more absorption can happen if there's more surface area. All right, so the next topic we are going to cover is going to be about DNA. So first of all, recall that you have a sperm cell, meets with a egg cell, and the sperm has 23 chromosomes and the egg has 23 chromosomes, and then once this sperm fertilizes the egg, you get a zygote, and that has a total of 46 chromosomes. So each somatic cell in your body has 46 chromosomes, and because they're essentially coding for like the same protein, maybe just different variations and stuff, we say that you have 23 pairs of chromosomes, okay? Just like you have, you might have 46 shoes, but there's just 23 pairs, right? So that's sort of how they work. Just remembering that there are 46 chromosomes and that there are 23 pairs. Next topic is I guess within the same topic, we have the central dogma of biology, which basically tells us that you go from DNA to RNA to proteins. Now, if you want to make a copy of DNA, like just a whole copy of the DNA, like replicate the entire thing, well, when do you need that? First of all, when you go into mitosis, right before that, let's say the cell decides to become two daughter cells. Okay, well, she has 46 chromosomes, right? Like we just said. Well, you need to have, each one of these has to have 46. So you can't, there's no way to like divide, go through mitosis. Your daughter cells don't have 46 each, right? And of course, we're talking about mitosis here, not meiosis. So what she's going to do is basically replicate all her DNA. So now, making a copy of each DNA, each chromosome, you're going to end up having 92 chromosomes. And then you divide that, and each one of them get 46. So this, basically, long story short, this process of DNA making a copy of itself is called replication. And DNA is pretty stable. So in order for it to be stable, because you want it to be as stable as possible, because it's going to get passed down from one generation to the next offspring, to the next offspring, right? And it's been that way for so many years, and you want it to continue that way. So the DNA is stored in the nucleus, where it has an extra layer of protection from the oxidative molecules within the cell. And so when you want to make a protein, what do you want to do? Remember, the DNA has all the genetic instructions you could possibly need to make any protein that your body can make, right? And so what ends up happening is that you go and you take a copy of this DNA, and this copy now can come outside and find the ribosome and sort of make that protein. So this idea of going from DNA to RNA, it's called transcription, because you're still using the same exact language. So it's transcribing, right? But when you go from RNA to proteins, this is also called translation, because now you're using two different languages. This is a nucleic acid, this is proteins, two completely different languages, right? So that's translation. So understand transcription is DNA to RNA, translation is RNA to proteins. Now, looking at that a little bit closer, we said that this mRNA, well, the mRNA stands for messenger RNA, is the copy of the actual DNA, okay? So here you have a double helix DNA, that some portions of it open up, right? So that this special protein called RNA polymerase, whatever, can sit there and make a copy of one strand of DNA. Now, when it makes that copy, what you end up with is this structure right here, which is the mRNA strand. And it is single strand, right? And he can now come outside the nucleus and find a structure called ribosome. This ribosome has this portion on it, it's called the ribosomal RNA, right? This is where the mRNA is going to meet with the tRNA. So it's just a special location at rRNA is where the mRNA and tRNA are going to meet. Now, who's this tRNA? This tRNA has, think of it as an RNA, right, that brings in amino acids, okay? So because mRNA by himself is just a recipe, and the tRNA is basically a small portion of the recipe, right, with an amino acid attached to them. So if this region here is complementary to this region here, within their RNA, he's going to actually end up dropping off his amino acid, and that's exactly what you see here. So you see these have already been dropped off, these amino acids, but this portion here, this is, remember, the mRNA meets with the tRNA, and because they are complementary to each other, they stick to each other, he's willing to actually let go of this amino acid, and this is how you end up making one amino acid after another. Now, the first amino acid that's always made, well, I don't want to say always because it's biology and there's always exceptions, right? But most often, the first one that's always made is methionine. So this guy here, this first, I guess you could say codon, right, is coding, AUG is coding for methionine, okay? So the first tRNA, when this guy was back there, right, because he's kind of just moving along, but when he's back there, the first amino acid that you made, which is this right here, as you can see, it's methionine. So tRNA that's carrying a methionine is going to be your first sort of translation, for protein synthesis translation, right? So translation really takes place starting at like a methionine amino acid. So we talked about RNA going to proteins, that's called translation, and I think that's really the main things you need to understand from this. Yep. Okay, the next, let's go over cell cycle. So really quick, recall that you have basically two main phases of cell cycle. There's the interphase and there's the mitosis. And the interphase, you can think of it as where the cell grows, replicates, DNA, makes organelles and all that stuff, right? So interphase is actually broken down into three subcategories, subphases, I guess. There's a G1, right? G1 is just basically growth, right? So think about this as a baby cell. He's just growing in this phase, all right? Then we go into, then the cell decides, okay, you know what, it's time for me to have a daughter, right? The daughter side should start to maybe think about mitosis somewhere down the line, division, cell division. And so first thing he's going to need, or she's going to need, is going to be a duplication, sort of a replication, I guess, of the genetic codes. Remember, because if she's going to have 46, right, there's no way she can pass down 46 to this one and 46 to this one. So first thing she needs to do is replicate all her DNA, right? And this right here actually happens in the S for synthesis, right? So synthesis phase basically makes your, it replicates the DNA. And then from S, you go into G2, and G2 is just more preparation. And around each phase of these is like a stop and like a red light and like a green light. You can go, you can stop, make sure everything is correct, right? And that's essentially what that G1, S, and G2 do. Remember, these are all part of the interphase. So far we talked about. Next phase is the mitosis, and mitosis is literally that time that the cell goes through division, okay? So mitosis is cell division, and we're going to talk about it in just a second, how it's made up of prophase, metaphase, anaphase, and telophase. I'm going to talk a little bit in depth for each one. I think it's beneficial for you to understand it, both for lab and lecture. So after mitosis, again, it goes through the G1, S, G2. Now, not all cells keep dividing. They're not all mitotically active, okay? Mitotically active means it just keeps going through the cycle, right? And you could assume, actually, that cancer cells are very mitotically active, right? They're constantly going through this. In fact, they're going through this without any, they're just blowing all the red lights, right? There's no sort of stopping for them, right? But in contrast, you have cells like in your spine, your spinal cord, you have neurons. And these neurons have, they're not mitotically active. Because they're not mitotically active, once they're done growing a little bit, so maybe perhaps after G1, right? They do not synthesize their DNA because they're not needing to go through mitosis. So after that, they just basically go through what's called a G0. This G0 or G0. Not something you need to understand, but I guess it helps to put things in perspective, right? So if a cell is in this phase right here, the G0, I guess I should have called it not G0, it's not mitotically active. It's just kind of dormant. He's living there, but he's not going to have a daughter. So she's not going to have a daughter. So let's talk about mitosis now. So before we actually do that, let's go ahead and look at the difference between chromosomes and chromatids, which I often see people confused, and including me, I used to always get confused with these things. So we said that the cell has 46 chromosomes and 23 pairs of chromosomes, but for now, let's kind of forget that there's such thing as pairs of chromosomes. When we're talking about, the mitosis, it's a little bit easier. Okay, so we have a total of 46 chromosomes. This is like in G1. Okay, so this is before mitosis even happened. So instead of G1, I could have put before mitosis. So you have 46 chromosomes and 46 chromatids. Now, notice, once you go through that S phase of the interphase that we talked about earlier, essentially what ends up happening is that this chromosome is going to replicate. It's going to make another version of itself, and they're actually going to sort of bind to each other in this fashion, all right? And there's this little thing in the middle here. It's called the centromere. And this centromere sort of holds these, and they all have this little centromere if you really want to be specific. And it sort of, centromere acts to hold these chromatids together. Now, so a chromosome is any one of these thread-like structures. Now, after you replicate the chromosome, right? What ends up happening is that you don't get 92 chromosomes, all right? You don't get 92 chromosomes. Why? Because they tend to actually stick together like this fashion right here. And because of that, you get 92 chromatids. So each one of these is a chromatid, right? Now, because this one here by himself, we say, yeah, it's 46 chromosomes. We have 46 of these guys, right? I just drew one. But as far as chromatid goes, we have 46 of them. So 46 chromosome, 46 chromatid in G1. Now, if you look at after S phase, because they stick together, there's still 46 of these chromosomes. But each chromosome is made up of 1 and 2 chromatids, right? And because of that, we say there are a total of 92 chromatids. So these guys here, like these two, the two chromatids in each chromosome now, and we call these sister chromatids, right? And so looking at the mitosis real quick. So notice this is prophase. Actually, both of these phases are prophase. And what ends up happening in prophase, here they have a schematic of both early and late prophase, but really just the same thing for our purposes. We have in prophase, we have the nuclear envelope sort of breaking down. And when you get this, you have the chromosomes going all over the place, right? And notice these structures, these black dots here are your centrioles. And remember what centrioles are made up of, remember microtubules, but what they make are these spindle fibers, these sort of fibrous things that come and grab on to these centromeres. So these guys here, actually, because there's a lot of mirrors and central and stuff, so let me write it down. So these are called centromere. And these are centrioles, so make sure you know these centrioles. And of course, these red lines are the spindle fibers. And yeah, so in prophase, you get the breakdown of the nuclear envelope, and you get the centrioles to move to these poles, these ends of the cell, and they start to throw down their spindle fibers on to grasp the chromosomes there. And then notice that each one of these chromosomes here, again, it has there are 46 chromosomes, but there are 92 chromatids. Okay, so throughout the cell cycle, you're not, even though that I mentioned that the mother cell, I'll put mother cell, right, becomes two daughter cells, and each one has 46 chromosomes, all right? The mother cell, she has 46 chromosomes before she replicated her DNA, but even after replication, she still has 46 chromosomes, but she has 92 chromatids. This is chromatid and this chromosome. So try to realize this. Okay, try to, I guess, picture it. So after prophase, we go to metaphase, and metaphase is just when all these chromosomes, along with their chromatids, right, chromatids, whatever, line up in the middle, called the metaphyseal plate. Just remember that metaphase is for middle, okay? So prophase and then metaphase, these are the two we talked about so far. And then next up we have anaphase. Now notice, in anaphase, you actually pull apart these sister chromatids. Now because you're pulling them apart now, they're no longer going to be together, and so each one of them is called a chromosome now. All right. So because they're no longer joined together, each one, you still have 92 chromatids, but now you have 92 chromosomes as well because they're not stuck together anymore. So until I guess before anaphase, you had 46 chromosomes and 92 chromatids, and then after anaphase, you're going to have 92 chromosomes. Anaphase and after 92 chromosomes and 92 chromatids. Okay, and then this is also another picture of the late anaphase. So these are both pictures of anaphase. And notice that these spindle fibers that are made by the centrioles are pulling the chromatids or the chromosomes to different sides, to the ends of the cell. And then in telophase, you notice that the nuclear envelope is created again, and really the spindle fibers have shortened, and they actually pulled the chromosomes towards the opposite side poles of the cell. Actually, fun fact, it's not the spindle fibers that pull them up, it's the chromosomes that move up. They sort of just like pull themselves up on the spindle fibers. But anyhow, and that's for telophase. So after telophase, our PMAT, prophase, metaphase, anaphase, telophase, has reached its end. So basically, we can say that mitosis has reached its end. Starts with prophase and the telophase. And one thing that I want to kind of mention in telophase is that you're starting to form, if you notice, two different cells. Like you could see that, right? I mean, in anaphase, even it looked like it was just one cell, but towards telophase, you start to see that they're sort of dividing. And this region right here is called the cleavage sphere. I think that's how you spell it. Yeah, so just keep in mind about the numbering. And then, of course, after mitosis has ended, then you have these two separate cells. This phase right here is called cytokinesis. Cyto means cell, kinesis means like movement. So the cells are ready to move apart from each other. And here again, each cell, since they're considered two different cells now, is composed of 46 chromosomes, and you could say 46 chromatids if you like. Okay, so keep that in mind. So that throughout mitosis, throughout the cell cycle, for most part of the cell cycle, you are actually going to at least in prophase and metaphase, you are going to maintain this the same number of chromosomes. Okay, only once you hit anaphase will your number of chromosomes change and become 92, just because they're no longer bound together through this my pen just died. They're no longer bound together through the structure called the centromere. And I think that's all I wanted to mention about this part. All right, so the next thing is cell junctions. There are three cell junctions you should know. First one is tight junctions. We already talked about this. This is to prevent sort of solutes and solvents from leaking between the cells, right? So you can imagine that where would you have a lot of tight junctions? Well, think about the skin, right? I mean, your skin, you can't absorb water through your skin, right? And that's because thanks to these tight junctions. So next one is gap junctions. Gap junctions, they're mainly there for cell communication. So there are these little protein channels. So here's one cell membrane, here's another cell membrane, right? So you can just imagine they're like this, and then there are like these gap junctions that are in the middle, and their main purpose is that, like, let's say some small polar molecules want to travel from one cell to another. They can do that by moving through these gap junctions. And these molecules that we're talking about, these small molecules, a lot of times they act like they are communicating molecules, right? They can depolarize a cell or create an action potential or so many different things, right? So that's for gap junctions. You can imagine that the heart has a lot of gap junctions. We'll actually get into that in just a second. The last one, that's up a cell junction, is the desmosomes we need to understand. And so the desmosomes, I would say, on this, it would probably be these here. Notice how they're kind of connecting all the cells together like this, and they're wrapping around so that nothing can actually pass. I'm trying to see if that's actually the desmosomes or not. But anyway, they wrap around the cell. So you have one cell here or one cell here, and they have these intermediate filaments. And what they do is they take these intermediate filaments and they go around the cell and kind of make sure everybody is connected to each other. So that if I'm pulling on this cell, it's not all the compression and tension is not just on this cell, but rather it's getting distributed through all the cells, right? So where would you think you have a lot of desmosomes? Well, your skin again, right? Because if you pull apart your skin, it's not just those cells that are feeling it. It's all the cells that kind of take on some of the word is tension, so that you're not that cell is not feeling everything. And you might also hear the word hemidesmosomes. Hemidesmosomes are basically the same thing. You don't need to know this, but if you hear it around, it's basically just cells, right? Remember, desmosomes are like attaching the cells together like this. But then hemidesmosomes, they attach it to the basement membrane, like they anchor the cell down, basically. In other words, and we're talking about mostly the epithelium. Okay, so far we've been talking mostly about like cells and like a little bit about organelles and macromolecules. Now we're going to talk about tissues. Remember that we have four types of tissues. You have epithelial tissue, connective tissue, muscle tissue, and nervous tissue. We're going to go in depth with each one. So epithelial tissue is up first, and one thing you should understand about epithelial tissue is that it covers all the exposed surfaces, okay, of the body. Now, I'm not talking about just like the external, but I'm also talking about internal surfaces of the body. So this is like your cavities, I don't know, around your organs, inside your blood vessels, right? So if you imagine you have a blood vessel, in the inside of these blood vessels, you have all these epithelial tissues, special types we'll talk about, but they're everywhere basically. In the gastrointestinal tract, all over you have these epithelial tissues, okay, inside and outside, really. So they're everywhere, okay, and their main job is to, well, they have a few main jobs, but essentially some of their characteristics is that they're very easy to regenerate, okay, so your skin, for example, right, all the dust you see in your room, it's majority of it, I think something like 90-something% of it is your dead skin, okay. So they have high regeneration. They also die pretty fast, but because of that, they have high regeneration. And because they have high regeneration, they actually are the first ones they are the most common ones to become like cancerous cells or like tumor cells, right? Because the more you replicate the DNA, the more likely you are to make mistakes, right? And so for that reason, a lot of cancers are epithelial derived cancers, okay? And they are avascular, which means there's no blood, but there's no direct blood vessel going to the epithelial tissue. And so they have to rely on diffusion to get their nutrients. Another thing that makes them super special is that they're very polar. Now, what I mean by polar is that polar means ends, right? So like the North Pole and South Pole, right? These are two completely ends of the Earth. And so if you look at like the epithelial tissue that I try to draw here, you could see, first thing you should always realize is this basement membrane area. And I put basolaminal, there's also basal, like reticular and stuff like that. These together come to form, it's called the basement membrane, sorry. So the basement membrane is this region down here where the epithelial tissue is anchored down to. And you notice that you have the epithelial cells right on top of it. And on this side here, it's called the apical surface. Now, this apical surface could either be, I don't know, the inside of the blood vessel, or it could be the inside of the intestine, or like the outside of your skin, right? So the actual environment that's outside. So these make them polar. That means they have two sides to them. There's the bottom side and there's the top side, the side they're exposed to, right? The apical surface is where they're exposed to. They are highly innervated, which means a lot of neurons go in that area. There a lot of sensory neurons are there, to say the least. And they have specialized contacts that we talked about earlier, tight junctions and desmosomes. Tight junctions to prevent fluid leaking between the cells, and desmosomes to kind of keep them all together, right? Some other functions, of course, is to protect underlying tissue, right? We're talking about physical barriers, physical protections, and absorption if we're talking about the GI tract, right? If we're talking about glands, that'll be secretion, right? If we're talking about the fact that they're very innervated, they do provide sensation in that sense, right? So the tip of your fingers are very, very sensitive because they have a lot of sensory nerves there. So now let's get to like the bread and butter of epithelial tissue, which is the cells that make up it. Okay, the cells that make up epithelial tissue, they come in four types. There's squamous, cuboidal, columnar, and transitional. Now, for these first three, you have simple or stratified. So simple means that if you look at the basement membrane, all right, there's just one layer of cells that are on top of this basement membrane. Stratified means if you look at this basement membrane, there's not just one layer of cells that are on top, but rather two or more, okay? So you can see that only one in the simple, there's only one cell that's touching, right? And in the stratified, one cell is touching the basement membrane again, but there are cells that are on top of it, okay? And so not all cells are touching the basement membrane. And so we're talking about squamous. Okay, so squamous, I always think about them as like being the most cheapest, like almost like plastic, right, to make. And so this means that if you lose like a plastic bag, you're not going to cry over it, right? If you lose a leather bag, you might. And so your body thinks the same way about this squamous because it's cheap. It tends to put it in areas that are like highly abrasive, I guess is the word. So like your skin is made up of stratified squamous, right? So many layers of squamous cells on top of this basement membrane. And then simple squamous is found in areas that you need like high diffusion. So think about your lungs where the gas exchange happens, right? Oxygen needs to come in and carbon dioxide needs to get out. Who better to put there than somebody who's very thin? Because literally the word squamous means thin. And they look like little fried eggs. And if you look at squamous, in the sense of stratified squamous, we have two types. You have non-keratinized and you have keratinized. I think about this like this. Look, non-keratinized is anywhere that dries if it's not wet. So what are we talking about? We're talking about like mouth. Like my mouth is pretty dry right now. It's because it's not wet. And that's because it's made up of non-keratinized stratified squamous. And same thing with vagina and esophagus. Keratinized, one of the functions of this keratinized cells is to make it water resistant. So you will find the skin to be made up of keratinized stratified squamous epithelial tissue. And that's skin everywhere, right? The palms of hands, soles of feet, most places, right? So again, it's water resistant, and that's different from waterproof. So it's not waterproof; it's water resistant. Okay. The next one is cuboidal. And cuboidal, it looks like cubes, so it literally looks like this. And simple cuboidal you can find in kidney tubules, cornea, salivary glands. Kidneys would be the best, like most textbook example of simple cuboidal. And stratified cuboidal, I would say glands in general, not all of them, but because it's pretty rare, I would say some glands are made up of stratified cuboidal. Now we're talking about columnar. Columnar, they are taller than they are wide, and so simple columnar is found in the digestive system, particularly in the small intestine, and their job is for absorption. And remember, it's simple because it's just one layer of cells on top of the basement membrane. And then stratified columnar, I haven't really heard many areas that I see stratified columnar, so they are rare again, and so I guess they're found in the anus and urethra if you really want to know. But one of the main ones to understand is this third type of columnar, which is called pseudostratified columnar. Recall that for it to be simple, all the cells need to be touching the basement membrane. If it's stratified, not all the cells are touching the basement membrane. And so if you look over here, here's the basement membrane, and it looks like there are cells on top of each other. But if you really look closely, each cell is touching the basement membrane, right? Each cell has a connection to the basement membrane, and this makes it pseudo-stratified. Pseudo means false. It's falsely stratified, which means it's actually not stratified. So a perfect example of this, pseudostratified epithelial tissue is found in the trachea. And more specifically, these cells have these structures called cilia, so they are called ciliated pseudostratified columnar epithelial tissue. So they're found in the trachea and nasal chambers. The next, last type of epithelial cells you have is called the transitional. So the transitional epithelial, there's no simple and stratified with this one. I'm assuming they're all stratified, kind of. So transitional, it's called this because it transitions from this form, looking like a cuboidal structure, to a squamous looking structure. So you find this in the bladder. So anywhere that can really go through sort of expansion, or needs to go through a lot of expansion, right? So the bladder, when you're relaxed, when you don't have to pee, there's not any urine in there or not much urine in there, it looks something like cuboidal. And then when you really have to pee, so it's really stretched, then it starts to look like it's squamous, okay? And also in the uterus, where the baby is, I guess, made, and well, I guess not made, but you get it, just it's growing. So the next type is the muscle tissue. And the muscle tissue, its main job is to contract. And anytime it contracts, the cell shortens. You could see that for yourself if you flex your biceps or something. Muscles are highly vascularized, means they have a lot of blood going towards them, which makes sense because they use a lot of ATP. And the best way to make ATP is when you have oxygen, basically. You get the biggest bang for your buck. So yes, muscles are made up of actin and myosin, something we'll talk about when we talk about the muscular system, but probably not very important right now. There are three types of muscle. There's skeletal muscle, smooth muscle, cardiac muscle. So skeletal muscle looks pretty cylindrical. Smooth muscle looks like a spindle shape, and this is just one muscle cell for each that I'm drawing here. And cardiac muscle looks like a branched, I don't know, like a tunic fork looking structure. The skeletal muscle is voluntary, of course, because it's attached to your let's say the biceps brachii, right? He is attached to the ulna, as well as his origin is attached to like the humerus. And so when you contract him, and you do this voluntary, what you end up doing is you shortening him, and actually maybe I should have drawn the ulna. Yeah, so the ulna becomes closer. So basically you're contracting him, and you're doing this voluntary. And if you look at the muscle cell, it has many nuclei, right? So one muscle cell, one myocyte, has many nuclei on it, so it's called multinucleated. And this is important if you were ever wondering why there's such thing called muscle memory. In a sense of like if you lose muscle and because you stop working out and then you go back to the gym, even though you haven't worked out for like 2 years, you end up gaining all that muscle back really quick. That's because although you didn't make new muscle cells, you went through hypertrophy and you made extra, you made the size of the cell bigger. Also, you added more nuclei to it. And so because it has more nuclei now, it can go through that translation process, I guess the transcription process faster, right? And so it has striations. These are the striations that we're talking about. And it is, yes, long cylindrical and it attaches to bones. Smooth muscle is the next one. So understanding that smooth muscle is found in a lot of these hollow organs, like the gastrointestinal tract, the blood vessels. Of course, it's involuntary. You don't control where your food is. You don't control your peristalsis, right? It's also found in the uterus and the urinary bladder, yeah. It is single nucleated, so it only has one nucleus, and it's actually called uninucleated, I believe. Anyhow, it's non-striated. It's the only one that's non-striated. And then we go to cardiac. And cardiac muscle is, of course, involuntary. And where is it found? Well, cardiac means heart, so it's found in the heart only. And it's involuntary. It is single, sometimes like it's uni or dinucleated, I guess. But for our purposes, we stick with one nucleus, okay? It is striated, so these are the striations you see on him. And it has this special thing. He's the only one that has this thing called intercalated discs. So these intercalated discs, if you look, so here's one muscle cell. But if I were to draw like 3 of them here, you could see that when they are touching each other, and this little black, I guess, darker region appears under the microscope, from where they're touching each other, and these are called intercalated discs. What do you think they're made up of? Gap junctions. Gap junctions, because these guys talk to each other. So he's like, I'm going to depolarize and I'm going to contract. And he tells him this, and he's like, well, you know what? I'm going to do the same thing. And he tells him this, and he's like, you know what? I'll do the same thing. And so this is how they're able to actually, your heart is able to like contract very like synchronously and without the need of your nervous system. So yeah, made up of gap junctions, intercalated discs. That's for cardiac. Skeletal is voluntary. It is multinucleated. It's striated. So is cardiac. Cardiac is also striated, but of course it's involuntary. And smooth, it is non-striated and involuntary, and you found them in these regions. Make sure you know these things. So the next type of tissue is nervous tissue. Nervous tissue is made up of two things: neurons and supporting cells called glia. So of course we all know what neurons look like. Here's a textbook picture of a neuron, and notice that the neuron has these things called dendrites. This is where the signal comes in. The soma, the body, where the I guess you could say whether it decides if the action potential is going to happen or not, right? Of course, the soma also contains the nucleus and all that stuff. This is actually where the action potential decides whether it's going to happen or not. But anyways, then you have the axon. The axon is this long sort of structure for most of them where the action potential is generated and passed through. And then axon terminals, this is a region right here where you have the these sort of synaptic vesicles that are packed with these vesicles are packed with neurotransmitters. So that's for neurons, and their main job is, of course, to create nerve impulses. Okay, and there's different ways to categorize neurons. Now we're talking about nervous tissue and not the nervous system. But when we get to the nervous system, you see there's categorization based off of shape, function, where they're located, whether they're inhibitory or excitatory, and all that good stuff. So the glia are the supporting cells of nervous tissue. So the reason why they call glia actually means glue. So back in the day when they were doing experiments on nervous tissue or neurons in general, they were noticing that there are these like glue-looking stuff around them, and they're like binding to them, and every time they have to kind of peel these annoying glue-looking things back just to get to the neurons so they can study them. And they were like, you know what, let's just call these things glia, right? And so the name stuck. But now today we know that they're actually more important than the neurons themselves. So that's I really hope this is recording. Okay. So that's the purpose of the glia. They act as housekeepers. Okay. So the main purpose of glia is to support and insulate the neurons. So without glia, you could say goodbye to neurons, because neurons, they don't know how to take care of themselves, okay? And so there are different types of glia, and I categorize them into macroglia and microglia. A few different types of macroglia, I guess. You don't have to call them macroglia. And you don't have to know this probably, but I'm just going to mention it anyways just to make sense. You have ependymal cells, and these ependymal cells, they produce this thing called cerebral spinal fluid. So if you ever look at the brain, try to draw a brain, it's terrible, but here's a brain. Yep, as best as I could get. Okay, so yeah, that's a brain. But if you ever notice, there's this like hollow structure inside the brain. These are like the ventricles of the brain, and it kind of goes down, it goes to the spinal cord, and all the way down spinal taps and stuff that you heard about. So there's fluid in these ventricles, and these are all thanks to these ependymal cells that make cerebral spinal fluid. Anyhow, the next type are the astrocytes, and these astrocytes are around the blood vessels that are in the brain, and they make sure that blood doesn't kind of like leak through the leak outside the capillaries and into the brain. They're confined within the blood vessel, and whatever nutrition that the brain needs will kind of be exported through these like astrocytes and stuff like that. And then there's oligodendrocytes. Oligodendrocytes and Schwann cells, they both produce this thing called myelin. So on the axon, you're going to have these like little structures that are like insulators kind of, and they help push the action potential down faster. That was a Schwann cell I just drew. That's in the peripheral nervous system. And then you have oligodendrocytes. Those are going to be in the central nervous system. But they do the same exact thing, made up of different types of myelin, but still the same thing. And the last one is microglia. Microglia are the immune cells of the central nervous system. Recall that you have this blood-brain barrier, and which means a lot of cells cannot get out. A lot of things cannot get out of the blood, including some of the white blood cells. And so these microglia are special types of immune cells that are in this central nervous system. All right, last but not least, connective tissue. Connective tissue, everything we didn't talk about falls into their connective tissue. So they are the most abundant. They're there to support, insulate, bind, just like what connective means, transport, store. We talked about protect already? Yeah, protect and defend. Okay, the body, I guess you could say. And they are all derived from the mesoderm, right? So there's the ectoderm, mesoderm, and endoderm, and these are the three germ layers. But essentially, they come from this mesoderm, and more specifically, they come from these like mesenchymal stem cells that are that come from the mesoderm, right? So the stem cells themselves are called mesenchymal stem cells, and they come from the mesoderm. So they have, of course, like the cool thing about connective tissue, just to kind of step back a little bit. So all those tissues we talked about so far, we're really just talking about cells, right? We were like, oh, here's the tissue, but it's made up of this and this and this cell. But now we're talking about connective tissue, and that just takes a turn. So connective tissues are not just confined to the cells they're made up of, although they do have specialized cells, but also their extracellular matrix. Okay, this is actually the main determiner and like the factor in determining what kind of tissue you're going to end up having, what kind of connective tissue you're going to have. And so what is this extracellular matrix? It's made up of this thing called ground substance, and ground substance is basically fluid. with some proteins, all right? So many different types of proteins. We're not going to get into it. But, so it's just like viscous, gel-like fluid, all right? That's what the ground substance is. Another thing that makes up the matrix are these fibers. And these fibers, we're talking about collagen fiber, elastin fiber, or reticular fiber, right? And of course, we talk about specialized cells. So what do we mean by specialized cells? We are talking about fibroblasts, right? These fibroblasts are the ones that make up the fiber, right? So all these fibers I just mentioned are made by different types of fibroblasts. And this is, they're located in the connective tissue proper, which we'll get into in just a second. And you also have chondroblasts. Chondro means cartilage. So chondroblasts are, cells that are in the cartilage. And then osteoblasts, osteo means bone, and hematopoietic stem cells, right? Hemato meaning, blood. So this is hematopoietic stem cells. They're found in the bone marrow, and they are what become the blood cells. Now notice that I mentioned blasts. Blast just means like immature. Oh, I guess not this. But blast means the immature form of a cell, right? And then mature form of it. So for example, osteoblasts will later on become osteocytes. Okay, and same thing with chondroblasts, they become chondrocytes. Usually, like osteoblasts, once they build bone, right? And then once they're sort of trapped themselves inside the bone, they kind of mature and become osteocytes. That's that for there. Okay. Yeah, there are other types of cells too. There are like fat cells. We call these adipocytes, right? They store nutrients. There are white blood cells, which is also called leukocytes. Leuko meaning white. Those are composed of neutrophils, eosinophils, lymphocytes, and their main job is to respond to infection and injury. There's also mast cells. Mast cells, their main job is to hit the alarm system. Basically, they have like an internal alarm system. Once they hit it, they produce a bunch of histamine. And if you don't know about spring allergies, well, let me tell you, it sucks because you have like literally structures like particles like pollen that's not going to even hurt you or harm you, but when I sniff it, my body thinks it's going to like kill me, and so it starts releasing a whole bunch of histamine, and then my eyes get all watery, my nose starts running, and it's just terrible, right? And that's just like allergies are like false signals, right? So yeah, these mast cells, inflammatory responses by releasing histamine. Now what histamine does actually is that it dilates. So the whole purpose of these guys is that when they come across a bacteria or like an area that's infected, so it's like full of bacteria, they release this histamine in the blood. And so here you have a blood vessel, and once this guy starts putting histamine, releasing it, that area becomes large, like that. It goes through vasodilation, and vasodilation causes more blood flow in that area, right? More blood flow means more white blood cells are going to probably come in that area. Well, they're right because you're increasing the flow of things that come in that area. So one of which is going to be the white blood cells, and it's always going to signal for white blood cells to come. And once white blood cells come, they check it out and they're like, okay, well maybe there's there are these bacteria, let's fight it. So that's the whole point of this inflammatory responses. Okay, and then you have macrophages. Macrophages, same thing. They're like Pac-Man. Oops, that's a terrible Pac-Man, but they eat, right? And then erythrocytes. Erythro means red, and then cytes means cells. So that's the red blood cells. So getting into the bread and butter of these connective tissue. Types of connective tissue. There are different ways you could break it down. I prefer this way. It's how it stuck with me, but do it however you want. But make sure you actually break things down in your own way and sort of make like I guess a flow chart or whatever you want to call it, like a diagram, right, of everything. So connective tissue, I like to break them into proper, supportive, and fluid. All right. So connective tissue proper then further breaks down into two categories. That's dense and loose. And supportive connective tissue, it can be bones and cartilages. And connective tissue, fluid connective tissue is blood and lymph. Okay, so first we're going to talk about this guy right here. So first of all, why is it called connective tissue proper? It's sort of everywhere, but why do we categorize it into loose and dense? So loose is because it has a lot of ground substance, but very low amount of cells. Right, so it's low cell volume. And then dense, it has a high cell volume, but low ground substance. Okay, so what are the different types of loose that you have to know? Well, all of them, right? So reticular connective tissue is essentially there for support, and you find them in the lymph organs, spleen, and bone marrow. And it's called reticular because it contains reticular fibers. Yeah, let me make sure go back up there and actually talk about fibers real quick. So the three different types of fibers are going to be collagen fibers, elastin fibers, reticular fibers, and collagen fiber is the most tensile strength, the high tensile strength, so it's really strong, right? But it's weird because it's a little bit flexible too. So it's not terribly bad, but it's mostly known for its tensile strength. Elastin, on the other hand, it's highly elastic, right? So that means you could stretch it a lot, and you could also let it go, and it recoils back to the shape it was, kind of like a rubber band. You could stretch it, and then when you let it go, it recoils back to its original shape. And then there's also reticular. Reticular is sort of, I would say, in the middle of the two. And so it's actually made up of collagen fibers, but they're very branched and weird and stuff like that. We don't really put a lot of emphasis into it, but still, it's somewhere in between like this guy and this guy, I would say. So talking about reticular, where it's found, and then next one is adipose. So adipose tissue is made up of adipocytes, so adipose cells, and their main job is to store like triglycerides, right? And there are two places, there are two places you can store adipocytes or adipose tissue. There's like in the abdomen area, right? And then there's like subcutaneous fat, right? So fat that's under your skin. But both of the job of adipose tissue is to like insulate and like protect you, and also keep in there triglycerides. Remember, triglycerides are your long-term energy, right? This is your body stores them as a form of like long-term energy because they have 9 kilocalories, whereas like carbohydrates, they have 4 kilocalories, and we're talking about per gram, of course. And so it's more worth it to hold on to these because you could get more out of them. Excuse me. And then the next one is areolar. This one's a really important one because I always thought about them. Actually, let me show you the types of. So yeah, adipose tissue, I don't really like this picture as much, but because we usually, when we look at adipose tissue, we actually zoom in one more magnification. So you see like honeycombs when you look at adipose tissue. But the reticular is pretty spot on. You can see like the mesh of like these type 3 collagen network stuff. The areolar connective tissue has a lot of different fibers and a lot of different types of cells that are in there. And it's kind of cool because it's used as like a packing material, right? So right between the skin and muscles and other organs, between organs and within organs and all that stuff, like between blood vessels and like, I guess you could say capillaries, and like the outside, you'll always find some of these like areolar connective tissue because they also have some empty space, and this empty space is perfect to kind of hold fluid. But if you don't pull this fluid back in, if you ever notice, if you look at somebody who's like a little bit older, they have like chubby feet, right? That's called edema, right? So that's when you have like fluid buildup in the tissue, right? And that tends to be in areas that have areolar connective tissue. So that's where the edema most often forms. Yeah, so they're everywhere. They're on blood vessels, nerves, under the epithelial, and that's really it for loose. Talking about dense, remember dense has a lot of cells. So there are 3 types of dense. There is going to be regular, so dense regular connective tissue. That's referring to these structures where you have a lot of, like if you look at the regular, all the fibers are kind of going in parallel to each other, right? So it's pretty cool. And if you think about what's the importance of this is that these fibers are really good at sort of withstanding tension from one direction. Like I guess you could say, what is it? One dimension, I guess, I don't know. But imagine here's your muscle belly, right? And you have tendons on both sides. These are supposed to be the tendon. Well, tension is either this way or this way, right? And so since all these fibers are running this way, like up and down, it's perfect to, I don't know how to word this exactly, but it can withstand tension from one direction, basically, okay? Now, contrast that to irregular, right? So irregular, dense irregular connective tissue, and that looks like this. So the fibers are all over the place in different directions. So this one here, it can actually withstand tension from many different directions, right? So these collagen fibers are just arranged in different patterns, right? So you can resist, it offers resistance in different directions, basically. And where do you find regular? Well, we just said tendons, ligaments, right? And irregular is found in the dermis, okay? So these are not parallel, and they resist pulling from different directions. And then last one is elastic connective tissue, and that's basically has elastin fibers, I believe. I don't know why I put elastic fibers. Yeah, and that's basically makes it up. We don't really talk about this guy as much. Okay, next. All right, the next type, we are going into the supportive connective tissue. First one, let's talk about cartilage. So when you ossify or calcify cartilage, you end up making bone, right? That's like what arthritis is, right? And the main job of cartilage is to withstand compression. So compression is like pushing something, like compressing it, and then tension is like it pulls away, right? So it's withstanding compression and tension. Now what you need to know about cartilage is that it's aneural and avascular. Okay, so no nerve cells or blood cells. Contrast that to the epithelial, where it had neural, like it was very innervated, but it was avascular, right? So this guy is both aneural and avascular, which means that it really, really depends on diffusion to recover, right? This is why if you damage your cartilage, it's most often not going to recover that well. Talking about the cells that are there, they're called chondrocytes, and the type of matrix that makes up the cartilage determines what type of cartilage you have, because you actually have 3 different types of cartilages. You have hyaline cartilage, you have elastic cartilage, and fiber cartilage. So hyaline cartilage is basically going to be the most, it's going to be the most abundant one, okay? And it's there in areas that need some like cushioning, right? So in the cushioning structure between like long bones, so right here. Hyaline cartilage, you could see, you can find them between like here, between the long bones, right? There, right? All these places that have hyaline cartilage, the blue, okay? And also the rib cage, the costal cartilages, right, are made up of these hyaline cartilage. Yeah, the larynx, trachea, also the larynx is like your voice box area, the trachea is the wind tube, the sternal cartilage we just talked about, then the external ear canal, that's like right after your ear, like the canal that makes up that ear. And in the areas of long bones, like in the joints, their lubrication is dependent on the synovial fluid that's in that area. We'll talk about more about this later on. So elastic cartilage is basically, so hyaline cartilage is it's not, it's somewhere in the middle. So here's how you should think about it. Fiber cartilage, super tough. Elastic cartilage, super soft, as far as cartilages go, right? And then hyaline cartilage is sort of in the middle, okay? Now, we're talking about elastic cartilage. So this guy is, actually, pay attention to something else. Hyaline cartilage, if you ever look at the microscope, it has, of course you can't tell based off of the matrix it has, but if you look at the lacunae, it has, the lacunae is where the chondrocytes sit in. So it has a decent amount. Now, look at the elastic cartilage. It's blown up with all these lacunae, right? So you have elastic cartilage in the ear, nose, and epiglottis. When I say ear, I don't mean the whole ear. I just mean like the part that's sticking out, like the pinea, right? So that's the back of the ear that you can fold downwards, that area. Is elastic cartilage. And the last one is fiber cartilage. Now notice fiber cartilage barely has any chondrocytes, barely has any of these lacunae, right? These houses for the chondrocytes are called lacunae. And fiber cartilage is found in areas such as intervertebral discs, temporomandibular joints, the menisci of the knees, and the pubic symphysis. So I used to ask this question a lot in lab. What is the pubic symphysis made up of? And that's this area right here. Notice, have you ever heard of somebody like tearing this area or anything like that? It can happen during pregnancy, but it really doesn't often. That's because it's made up of fiber cartilage and it's super tough, right? And between these intervertebral discs are also made up of fiber cartilage. It's super, super tough, okay? They don't move as much, but also they're very tough, okay? Between dens, connected, yeah. Last type of supportive is bone. And bone, when we talk about the skeletal system, we talk about more about spongy and compact. But for now, just understand that bone is a type of connective tissue, of course, and it's also known as osseous tissue. It has cartilage that's ossified, but also calcium, like also inorganic salt, so like calcium phosphate, right? These structures that make it really, really tough, right? It makes it hard, basically. If we're talking about the compact bone, especially. It is highly vascular, of course, right? I mean, that's where actually the majority of your blood cells are made in. So, of course, the job is to protect body structures. It stores some fats in the yellow bone marrow and makes blood cells in the red bone marrow. Has collagen, more collagen than cartilage, and of course calcium salts. We talked about this, yeah. So osteoblasts, they build bone, and basically these osteoblasts sit, like you see these little lacunae that are here. They trap themselves in this area because they build bone all around themselves, and once they're done with building bone, they just become osteocytes. They mature to become osteocytes, and they still play a huge role. But anyhow, we're not going to get into that right now. Yeah, that's the lacunae I was talking about, and this is the osteocyte at this point. Right? And so, kind of understand that osteon, which is this whole thing that I circled, is the functional structural unit of a bone. Okay, and of course it's highly vascularized. I think I've mentioned everything I wanted to here. Okay, next one is blood. This falls into fluid connective tissues. It's blood, and blood is just blood, right? So it's made up of white blood cells, red blood cells, I don't know, platelets, all these guys. And it's surrounded by non-living fluid matrix, which is called blood plasma, right? Or plasma, when we talk about this. So, and then the next fluid is lymph. We're not getting into lymph as much, but it plays some immune functions. It has some lymphocytes, and there's a tiny little chapter about it in Anatomy 2. Okay, so next thing to talk about are body membranes that you have to know. There is, look into cutaneous, mucous, serous, and synovial. Understanding that cutaneous membrane is keratinized, and that's what makes up the dermis and epidermis. Mucous membranes are also called mucosa, and you could see they line the body cavities that are open to the outside, right? And so these are all the mucosa layers, mucosa membranes. So understand where they are. So what are the jobs is absorption, protection, and secreting stuff, right? Whether that's mucus or other things. And they're all over the place. They're digestive, respiratory, urinary, reproductive tracts. And then serous membranes, they are also known as serosa. And here, this is us dissecting a cat last semester, and I'll get into that in just a second. But they line body cavities, okay, that are not open to the outside. So think about your pleural, the thoracic cavity, or the abdominal pelvic cavity, right? And they really come in two forms. You can either have parietal serosa that lines the inside body cavity walls, right? That's around. So like example of this would be, you see like this right here, right? This whole thing is the parietal pleural. And then the visceral pleural is the one that's actually touching the organ itself. It's visceral. Okay, so that's basically the difference here. Yeah, the structure you see here that we have is the peritoneum. Okay, so that's for that. Yeah, I want to explain actually this word here, mesothelium. So you might hear the word endothelia, mesothelia, epithelia. They're all referring to like epithelial tissue. It's just depending on where that epithelial tissue is. Because remember, there's a lot of places that they could be inside, outside the body, right? If it's endothelia, they're referring to it as being in the blood vessels or blood vessels, right? And remember, this blood vessels are made up of these simple squamous epithelial. Mesothelia are in this case. Most often going to be made up of simple squamous epithelial. I'm not actually sure. Pretty sure it's stratified, but mesothelia are referring to these guys here, right? And then epithelial, if they talk about epithelial, most often they talk about the ones that are like exposed to like the outside environment. But yeah, so if you hear this word, these are different words, just know they're referring to the same type of tissue. Okay, the last one, synovial membrane, and synovial membranes are basically the surface of capsules of synovial joints. Right? So these synovial joints have these capsules, and inside they have this synovial fluid. And remember, the synovial fluid is what lubricates these cartilages, right? These articular cartilages. Yeah, articulating cartilage is another word for hyaline cartilage. Also, maybe something I forgot to mention. Okay, anyhow. Tissues repairs. Yeah, so understanding that the processes of tissue repairing go through inflammation. What is inflammation? What's the purpose? We kind of talked about this a little bit. Organization and regeneration. So step one is inflammation. That's the first thing that happens. Where we talked about histamine being released, for example, and that basically dilates the blood vessel and allows it to be more permeable to, I guess not more permeable, but brings more like white blood cells or blood in that area in general. The next step is, of course, organization, and that's perhaps making a blood clot or something like this. And then there's regeneration, and that's like basically just regenerating new tissues. Understand that inflammation, acute inflammation of molecular systems causes unpleasant symptoms, but they will get it. Okay. Yeah, so inflammation in general, just sort of understanding like what are the symptoms, right? So there's becoming hot, becoming red, swelling, having a lot of pain, and being like numb, right? But not just numb, but rather like it doesn't, your finger is not going to bend as well in this case, right? So that's really, these are from here on out, I think they're pretty self-explanatory. I'll just go through like the things you should know, so you can go back and review yourself. So the integumentary system is up next, and of course the skin is the largest organ. We talked about how it has a lot of tight junctions and desmosomes already because it's mostly made up of epithelial cells, and there are also accessory parts to the integumentary system. Those are the hairs, nails, sweat glands, and these oil glands, sebaceous glands. Understand that the integumentary system provides a barrier to the outside world. We're talking about barrier, we're talking about like it protects you from the outside, right? That's in the sense of physical, chemical, this biological barrier, right? And yes, doctors could use this as a visual indicator of your health. And dermatology, dermo means skin, and dermatology in general makes a lot of money. And also knowing the functions, protection, body temperature regulation. So yeah, like when you have when your body's hot, right? What happens is you have vasodilation in the skin, and this vasodilation allows more heat to dissipate from the blood vessels and onto your skin and out into the environment. Of course, sensation, because there's a lot of like free nerve endings and different corpuscles there. Metabolic functions, stick with vitamin D, although there are some other ones as well, but the main one is vitamin D. And the purpose of vitamin D is to absorb calcium. This is why they fortify milk with vitamin D, so you can absorb calcium. Blood reservoir, yeah, so you have about 5% of your total blood volume inside the skin, like within the skin. And then also there's excretion of waste. We're talking about sweating, right, salt and water, as well as some other nitrogenous waste like ammonia, uric acid, and all that other stuff. Yeah, there's also different ways to regulate temperature, not really super high yield, but kind of understand that insensible sweat and sensible is through that vasodilation. And of course, when you're cold, you're going to have vasoconstriction, right, not vasodilation. This is one reason I tell you to take hot showers at night, is because when you jump into hot water, your body's going to do vasodilation, and then you step out of the shower, you go to your room, and then it gets really cold, and then like a lot of the heat leaves your body, and your body just cools down very fast, even though it's like it doesn't make sense for why are you cooling down if you're taking a hot shower, but that's actually how you cool down really fast, and then you just get sleepy right after that. Anyhow, yeah, so make sure you understand that the skin, or the cutaneous, right, is made up of the epidermis and the dermis, and not the hypodermis. Epidermis, you have a couple layers there, and the dermis is underneath the epidermis. It's the layer that's pretty vascular. We'll talk about this in just a second. I want to see where this came up. Okay, yeah. So actually, let me just talk about it now. Or I guess that's up next, actually. So there are different types of cells that are in the epidermis. There's keratinocytes. They're the main ones, right? They're the majority, and tightly connected by desmosomes. Yes, desmosome is what keeps the cells together. There are melanocytes. These melanocytes produce melanin, and melanin comes in different shades. And melanin is like sort of like sunglasses. Think about it like sunglasses that the melanocytes produce, and they throw it in front of the cells so that the UV doesn't hit the DNA, because UV can actually break down your DNA. It can form mutations within the DNA, basically. And then there are also dendritic cells that are in the cell, and these specific dendritic cells are called Langerhans cells. Langerhans cells are the type of dendritic cells that are found in the epidermis, basically. And then there are tactile cells, also known as Meissner corpuscles, or Merkel cells, right? And these are there for like light touch sensation. They're sensory receptors that sense touch. And so you can see that here, for example. So you can see the tactile Meissner corpuscles. They are in this region of the dermal papillae. Okay, so let's skip this part. Okay, good. Now we talk about epidermis. Understand that the 5 layers that are in epidermis, there are 4 layers in thin skin and 5 layers in thick skin. So only stratum lucidum is actually in thick skin. And thick skin is only soles of feet and palms of hands, right? And so the 5 layers, I always think about it as come, let's get sunburned, right? And so the come stands for stratum corneum. So when you're looking at your skin right now, you're looking at stratum corneum, essentially all dead cells, right? And then you have stratum lucidum. That's only if you have if you're looking at your thick skin, which is the palms of hand and soles of feet. After the stratum lucidum, you go into the stratum granulosum. So that's come, let's get. So that's the get part, stratum granulosum. And here you have like cells that are turning to be dead. Because remember, the cells are getting pushed this way. I'm just covering it the other way. But and then after the stratum granulosum, you go into the stratum spinosum. There's the spiny layer, right? And then after that is the stratum basale. So stratum basale down here is where it all starts. So all these here, there are stem cells, and these stem cells, they're either stem cells when they divide. How do I say it? Like stem cells when they divide, they divide and they make one stem cell, become one daughter stem cell, and then the other stem cell can become whether it's a keratinocyte, which is for the most part they're mostly keratinocytes in that area, or tactile cells or melanocytes, right? And so all the stem cells are down there, and it just become different things, okay? And these melanocytes, is this a melanocyte? It's a tactile cell. Yeah, here's a melanocyte. These melanocytes, remember, they make melanin. Yeah, another word for stratum basale is stratum germinativum, and after they just basically move up slowly. Also knowing that right after stratum granulosum, so these here, 4 and 5, stratum corneum and stratum lucidum, they are dead. Stratum granulosum is where they're kind of transitioning to become dead. And also you find lamellar granules that basically have these glycolipids there in that area. So this slows down the water loss, so that cells are not losing a lot of water in that area. So that's also found in the stratum granulosum. Here's a picture of the melanocyte and also other cells. Yeah, so I just wanted to talk about the, I don't think we go over the dermis as much. But the dermis, yeah, so most things are happening in the dermis, and of course the dermis, it's made up of this papillary layer as well as this reticular layer. The papillary layer is just this portion right here. The papilla is like little nipples, and so it looks like little nipples. That's why they call it that. And it's made up of areolar connective tissue, all right? The reticular layer is made up of reticular connective tissue, right? It's for like support and stuff like this. And so that's what the 2 layers of dermis are. Here are the 5 layers of epidermis. Also notice here you have different, here's the hypodermis. It's not part of the skin, but hypodermis. And you can see a lot of fat cells here, right? Adipocytes in this area, adipose tissue. I guess pay attention to the hair follicle and the hair bulb and stuff like this really quick. Notice they all have these little pili. Also have these erectile pili muscles. We'll talk about this in just a second, actually. So there are 3 ways you can get pigmentation. There's melanin, keratin, and hemoglobin that affect the pigment of your skin color. And so melanin, of course, we talk about there's different shades. Me and everybody else have the same number of melanocytes, unless they have like, some sort of carcinoma. But we have the same number of melanocytes, but we just produce different. Like mine might not be as active as some other people, right? They tend to be like darker color. Okay, that's the whole idea there. Keratin, this is where the name carrots come from, because they are yellow orangish pigments. Yes, if you eat too much of it, you definitely turn orange. And keratin, if you ever see your mom says like, oh, eat carrots or something, it's good for your vision, right? Because vitamin A is good for your vision. And keratin is the main molecule in carrots, and that's a precursor to vitamin A. Hemoglobin, it is the like the protein on the red blood cells, right? And you notice if you ever get really, really hot and you have a lot of vasodilation, if you're lighter skinned, you tend to be like more reddish. Anyhow, so these are also important clinical applications. Understanding that cyanosis is, cyano means blue, so that's like you turning blue because you have low oxygen amount. And there's also pallor, Pallor is like becoming a pale color. That's because you have low blood or low blood pressure, or you're scared to death or something. You become pale. Erythema, it's literally eryth means red, so this can come from if your body's too hot. Same thing we just talked about. You have a lot of vasodilation in the skin because you want to get rid of the heat, and so your body's going to look like it's red. Hypertension, inflammation, allergy, all these things. Jaundice is a cause of like bilirubin. Like your liver's main job is to like break down one of the main jobs. The liver has like 200 functions, but it's supposed to break down red blood cells, and blood cells have bilirubin, and this bilirubin needs to be getting like rid of. And so if your liver doesn't do a good job at that, if you ever see like neonatal jaundice, right, that's that yellowish color of the bilirubin. It tends to grow around, and you can see it from like under, like it's like right underneath the skin. So the baby turns like a little yellow or something. And so they put them under light, visible light, and that's supposed to help with like the breakdown of that bilirubin. Anyway, too much information. Jaundice, yellow, okay, and that's because of the liver is not doing a good job. So of course bruises, blah blah blah. You can assume these yourself. Hair. So hair is made up of dead keratinized cells. It's everywhere, sadly, except for palms, soles, lips, nipples, and by nipples we mean like the areola, like the brown part, and certain parts of external genitalia, right? Some of the functions, of course, you want to understand these for your own sake. They're pretty sensitive, so but like it can warn you of insect on the skin. You protects you against like physical, to a certain degree, protects you against like physical trauma, I guess, for lack of a better term. I'm just going to read off this. Also, a little bit for heat loss, depending on what kind of hair we're talking about. Like cats, that's definitely true. But for us, true if it's coarse hair, right? I mean, I know I shave my head a couple times and it gets really cold right after that. Also, shields the skin, right? That's what the lashes are, right? They're like little hats for your eyes. And yeah, just understanding this right here, kind of what they do, each of them, not super high yield, but understanding the shaft is a portion that sticks out. The hair follicle, it's like within the epidermis. Oops, I went too high, but it's like it's this part here. And then you have the roots or the bulb, and that's this region right here. That's you still form new hair, right? That's where hair comes from. And then papillae are these like little regions right here. They get nutrition through blood vessels and stuff like this. Erectile pili muscles help. These are like the erectile because they're erect. Right? And pili muscles, these are what make you have goosebumps, basically. All right, and they are important for when you're like under, like if you're really cold and you're shivering, and also these erectile pili muscles activate. They do generate some amount of heat, so your body can stay warm to a certain degree. But nowadays we have jackets, so we don't really need these stuff. So yeah, once they contract, the hair also stands up, right? Because it's kind of attached to the hair, so you can see that. Yeah, types of hair. You have villous hair and you have terminal hair. Terminal hair is like the hair in your eyebrows and like in your head, and then villous hair is like that pale hair that nobody really notices there. That fine hair. Yes, drugs, of course, especially like chemotherapy and stuff. They stop they stop hair growth, and nutrition plays a huge role in there. Nails, understand them a little bit. You got the nail bed. That's where's the nail bed again? Here. There's the nail bed right here, and then so the nail bed is like like this, the epidermis is underneath the nail, right? So if you cut your nails too short, so you could touch it and it's kind of sensitive. And the nail matrix is the region that's in charge of growing the nail. Cuticle, we all know what that is, right? Also known as eponychium, ponchium. I think it's eponychium. Anyway, yeah, so that's the cuticle. That's the little, like the folds that we tend to push back. And then you have the luneal, which is the white portion here, right? And this is where most often doctors will look at. So speaking of doctors and homeostatic imbalances, try to understand that like yellowish nails mean it's fungal infection. Colonchial, it's iron deficiency, and these lines, the Bio's lines or whatever they're called, they can mean like serious health illnesses, right? Glands. There are two types of glands. There's exocrine and there's endocrine. Exocrine glands have ducts, and endocrines they tend to come ductless. And so exocrine glands they always project to like outside, not like outside your body always, but like they pour something like outside of a region. But I guess it makes better sense when I explain endocrine. Endocrine always pour into the blood, so the blood is their main job. So endocrine glands are like the pancreas has some endocrine functions, right? It makes insulin and different glucagon and somatostatin and different things, right? The thyroid gland has endocrine functions, right? But exocrine, for our purposes, we're going to focus on sweat glands. And so there are three different types. We're going to talk about holocrine, merocrine, and apocrine. Holocrine is the only one that, in order for I guess I'm going to call it juice for lack of a better term, to come out, in order for that to come out, the cell has to actually like burst, okay? And they all have ducts. And so once it bursts, it like the thing comes out. All right. So sweat glands, also known as pseudonephron glands, they're everywhere except for nipples and some parts of the external genitalia. there are two types. There's the eccrine, also known as merocrine, and apocrine. And of course, they're going to have some myoepithelial cells, just cells that contract to push the stuff out, right? To push the sweat out. And so merocrine, they work through ducts. Maybe I should just erase this part. Okay, so merocrine is the most abundant. Their function is for thermoregulation mostly, and they get activated by sympathetic nervous system. We talk about apocrine. Apocrine is mostly in the armpits, so it's called axillary or pubic area. That's the anal genitalia. And they have this like yellowish, milky, fatty substance, right? And then when bacteria like eat it, they tend to like it lead to body odor, right? So a lot of animals, not us, but like a lot of pheromones and stuff essentially come from these things. They are larger glands compared to the merocrine, and we also have modified apocrine glands. These are like the ceruminous glands. That's basically what creates the ear wax, also known as cerumen. And then we have mammary glands. Those are located in the breast, and they produce milk. So those are the type of modified apocrine. Understanding that apocrine and merocrine don't rely on bursting to excrete their stuff. Okay, while holocrine does. Holocrine, the whole gland has to break down, right? So it has to die before it comes out. So these are one type of holocrine is sebaceous oil glands. Sebaceous glands, also known as oil glands, they produce this thing called sebum. Sebum. And this sebum acts as like an antibacterial, it has bacterial killing properties basically. And it's oily, so it's what prevents your hair and skin from drying out. And it tends to be, if I have a picture of it up there, it tends to be always around, you can see this here, that is a sebaceous gland, so it's always around the hair follicle. That's really the main takeaway there. Okay, skin cancers, be very familiar with them. You have basal cell carcinoma, pretty common, low malignant, and then you have squamous cell carcinoma, second most common, but it can metastasize, but it's not as dangerous as melanoma. So melanoma, super dangerous, highly metastatic, right? And then it's very dangerous, of course, because it's highly metastatic. And yeah, they're like asymmetrical, basically. Okay, the next one is burns, and I think that's the last one, thank God. So the next one, burns, you have first degree burn, second degree burn, and third degree burn. First degree burn, just the epidermis. Second degree burn is the epidermis and the dermis. Third degree burn, you have epidermis, dermis, and even some portions of the hypodermis. So it's going to cause you no pain because you just done burn through all your free nerve endings and mines their corpuscles and other cells, sensory cells. You're definitely going to be very prone to dehydration. Even second degree burn, if you ever came back from beach and you got a pretty bad sunburn, you know that you need to drink a lot of water because it puts you in a vulnerable position as far as dehydration and electrolyte imbalance goes. Yeah, you're also going to need, most often, for third degree burn, you're going to need a skin graft. And there's like the rule of nines. I had a picture of it, but it's fine. We're not going to talk about it. Just understanding that what each one does, right? How far does it burn? So first degree, just epidermis. What does it look like? Red swelling, right? That should do the job. I think we covered everything. Yeah, melanoma, we talked about this. Also know that it's pretty resistant to chemotherapy. That's good enough. All right.